Hi guys, nucleic acids, quick revision. Let's get straight into it. Nucleic acids. First we're going to look at the structure. First we'll look at the structure of the nucleic acids, and then we'll look at the function. It's really going to be that straightforward. So we'll have functions down here. What we want to make sure is we cover the three main molecules that we need to be aware of: DNA, RNA, and ATP. Okay, so we need to have them on there at some point, including the structures and functions, and we need to have a little bit of a look at extracting DNA. All right, so that will be there. So we got functions, extracting DNA, structure. Let's begin then. The structure is all about, really, the main part of all this is the nucleotide, right? The basic building block of nucleic acids is the nucleotide. So we need to remember its structure. Okay, so we have a phosphate group. Phosphate group, we have pentose sugar. Keeping things general for now, and then we'll get a bit more specific with that. Pentose sugar. We have a nitrogenous base. Phosphate, pentose sugar, nitrogenous base. The phosphate is connected to the pentose sugar by an ester bond, and the pentose sugar is connected to the nitrogenous base with a glycosidic bond. Okay, so that's our basic building block: phosphate group, pentose sugar, nitrogenous base. Okay, so that's our monomer in a way. That's our repeating unit, and well, in most cases, ATP isn't a polymer. But yeah, okay. So now what we need to look at is how DNA are formed and how their monomers are slightly different from each other. Let's look at the pentose sugar aspect of this then. So the pentose sugar can be of two types, right? So from here we could go, we could say, right, let's have a bit of a closer look at the pentose sugars. The pentose sugars are of two types, right? So DNA is made of deoxyribose, or DNA nucleotides. Their pentose sugar is deoxyribose, and RNA nucleotides are made or have as their pentose sugar ribose. And I think we need to be aware of what they look like. Okay, so deoxyribose is a pentose sugar. So we just draw a pentagon with the oxygen at the top right there. I'm not going to draw all the hydrogens out, so I'm just going to draw the OH group there. Deoxyribose doesn't have an OH group at carbon 2, right? It does have one here, OH group there, and it has a CH2OH right there. It's a pentose sugar, so 1, 2, 3, 4, 5, that's fine. Okay, and that's deoxyribose. Yeah. Okay. Ribose, however, will be similar. So again, with that is common, but ribose will have an OH group there, and it will have an OH group there, and everything else should be the same. So CH2OH. Right, so that's deoxyribose, that's ribose. We can see the main difference is at the 1,2 carbon 2. Okay, right. So remember deoxyribose, this is the pentose sugar in DNA, this is the pentose sugar in RNA. Cool. The other difference is in the nitrogenous bases. Right, these are split up into two main groups, right? So you have the purines and you have the pyrimidines. Right, what I want you to remember, now these are nitrogen containing structures. They have ring, they have got like a hydrocarbon ring structure with which incorporates some nitrogen in it as well. I don't believe you need to know exactly what the structure is, but you do need to be able to identify purines and pyrimidines. So let's just put that stuff up here. So the way I remember it is small word, big molecule, big word, small molecule. Okay, so the pyrimidines are the ones which have one ring in their structure, and the purines have two rings in their structure, something like that. Okay, so the purines, and I don't remember this, I'm just going to check. I think the purines are A and G. Let's see if I'm right on this one. And therefore the pyrimidines will be the other ones. So A goes with T and goes with U, and we'll have the C that goes with the G, and I hope I'm right about that. Okay, so remember adenine, guanine, in terms of the pyrimidines we have thymine, uracil, and cytosine. Okay, and yeah, and I guess we could talk about the base pairing since we are here, but remember that because these ring structures incorporate nitrogen and there's also hydrogen there, nitrogen is partially negative. Nitrogen is an electronegative element, so it is partially negative, which makes hydrogens that it's bound to partially positive. And because these partial positive and partial negative groups are in these molecules, when these molecules come near each other, these partial positives and partial negatives actually can align with each other in a way that they can hydrogen bond. All right, so I'm sure you guys know this off by heart, but just so that it kind of ties into some form of explanation, the nitrogenous bases have the base pairing, remember, A to T or A to U, 2 hydrogen bonds, and G to C, 3 hydrogen bonds. Let's look at then the structure of DNA. DNA structure, right, so it all begins with our nucleotide, right? But nucleotides, remember, they're put together by condensation. So I'm going to just put that here. Condensation reaction. Okay, so when you put two nucleotides, or they're not going to spontaneously react, but two nucleotides, right, can be joined by condensation reaction. It's the phosphate group of one nucleotide joins with the OH group of the next nucleotide, and in that way we form a phosphodiester bond. That's important right there. Okay, so that's a phosphodiester bond. Phosphodiester bond. Okay. But this can happen many times. So we could even have one up there. Phosphodiester bond. We could even have one down here. Okay, so pentose sugar, pentose sugar, pentose sugar, pentose phosphate, pentose phosphate, pentose phosphate. So sugar phosphate backbone with the nitrogenous bases projecting away from the backbone. Okay, so I hope that that's clear in our minds. Okay, and the phosphodiester bond linking the nucleotides together to form a polynucleotide. Okay, but that's not where DNA structure finishes. DNA structure has a counterpart strand. It's got a complementary strand on the other side. So we would have a nitrogenous base there. That would be connected to a pentose sugar as well. But remember that the opposing strand is anti-parallel. So it's complementary, but it's anti-parallel. What that means is the pentose, the nitrogenous base is flipped by 180 degrees and the pentose is flipped as well, and the phosphate would be down there. Okay, so if that's a C, that should be a G, and there should be 3 hydrogen bonds between the two, and it's the hydrogen bonds between the bases, complementary bases, that hold one strand together with the other one, with the complementary one. Okay, so let's just finish off the diagram. So we'd have another nitrogenous base there, pentose sugar, nitrogenous base, pentose sugar, nitrogenous base, pentose sugar, and they would be attached with phosphodiester bond, phosphodiester bond, phosphodiester bond. Remember, it's the condensation reactions that allow that to happen. And when we look at the function, when we look at when that new strand of DNA is synthesized, we'll see how that is formed. Okay, so this could be A, this could be T, this could be T, this could be A, this could be G, this could be C. Okay, 2 hydrogen bonds, 2, 3, and it's not clear, but you get it. Okay, so there we have that. So we have phosphodiester bond, and you can and should try and label these things, like know the names of what we're talking about here. So that is a sugar phosphate backbone, just the pentose and the, actually it's just that part there, sugar phosphate backbone. These are the nitrogenous bases. That's not new. Anything, you know, the idea that the two strands are anti-parallel. Did I spell that correctly? Probably not. Okay, the two strands are anti-parallel. Sugar phosphate backbone, hydrogen bonds, complementary bases, all that stuff. Still not done in an aqueous environment, right? While the phosphate groups, they're negatively charged, and the sugar molecules are polar, the nitrogenous bases are not polar, they are hydrophobic. So what happens, because all these bonds are flexible, I don't know if you've ever kind of played with a model of DNA, but the molecule has the ability to twist, so that the polar parts or the hydrophilic parts of the molecule can interact with the water on the outside, but the bases are kind of protected from the aqueous environment, kind of like a hydrophobic core. But in doing so, what happens is that this molecule twists and forms the very famous DNA double helix. Okay, so if you look at like a real three-dimensional rendering or model of the molecule, you'll see that the phosphate and sugar, you almost can't see the bases because the sugars and phosphates are are on the outside in that fold, in that coiled form. And there we have double helix, right? 2 strands of DNA, or 2 polynucleotides wrapped around each other, double helix. Okay, not much really to say about the RNA structure, not that there isn't, I mean, not that it's not interesting, it's just that structurally speaking, so if we go from here and we just go, right, let's say now we're looking at RNA structure. The RNA structure, there isn't really not a lot going on because it's single-stranded, right? So just remember that. So the DNA is a double helix, but the RNA is single-stranded molecule, so it would just have one of these. And it would carry on, but there's no counterpart complementary strand. Okay? Does that mean it's not capable of base pairing? That's not true either. It can base pair, but it's usually temporary and it's usually with a DNA molecule on the other side. Yeah, though there can be double-stranded bits of RNA as we know from tRNA structure. Okay, so there we have the structure of DNA and RNA. Would it be wrong of us not to do ATP while we are here? I guess it would. So I'm just going to put ATP here. Right, ATP structure. It's getting a bit messy. But ATP structure, not anything more complicated than, instead of just having one phosphate group, it has 3 phosphate groups, but it's the other 2 phosphate groups are attached to the first phosphate group. Yeah, so phosphate, so we have the pentose, which is ribose, right? So we have the ribose as the pentose sugar. What happened to my ribose there? Kind of melted, right? And we have the nitrogenous base, which is not any of the 4, but it is adenine, hence the A in the ATP. Right, so we have ribose, we have adenine, and to the ribose we have a phosphate group, to that phosphate group we have another phosphate group, and to that phosphate group we have another phosphate group attached, hence ATP, adenosine triphosphate. Okay, and that structure is important because the addition of those 2 phosphate groups, essentially that's where the energy storage property of ATP really resides in that structural element there, or in that structural bit. Okay, let's move on then. So let's now look at the functions of the nucleic acids. All right, come on, let's speed this up. Right, so functions, the main thing, well, shall we just make a little bit of sense of this before we move on? So we said we'd look at nucleic acid structure. From structure, we looked at the nucleotide as the basic and common unit. Part of that, however, we can have different pentose sugars. So we looked at deoxyribose and ribose. We can have different nitrogenous bases. So we looked at the purines and pyrimidines. Then we looked at the structure of RNA, DNA, and ATP. And now we're just looking at the functions, and that's primarily going to be DNA and RNA. Okay, and we'll spend a little bit of time on ATP, but not too much. Right. So let's begin. First main function of these nucleic acids is DNA replication. DNA replication. Okay, so there is a bit is the mechanism here. So the process by which DNA replication occurs is the semi-conservative model. Okay, so the semi-conservative model, so there's a process that we'll discuss, but also in relation to this, there are some experimental demonstrations which tend to come up quite a lot in the exams, even though it's not specifically stated on the specification. Okay, so it is useful to know the Meselson and Stahl experiments. Okay, that kind of provided evidence for the semi-conservative model of DNA replication. Okay, so what is a semi-conservative model? I don't want to go into too much detail, but I'm just going to skim through it very, very quickly. Remember, the semi-conservative model is all about step 1, helicase, the enzyme helicase unwrapping the double helix, unwinding, just going to put down keywords here, I guess, unwinding, unzipping, right, exposing those nitrogenous bases, right, exposing those bases. When those bases exposed, free nucleotides can align. The free nucleotides can align, right, and as they do so, they can get joined up as by DNA polymerase kind of moving along the molecule. So free nucleotides align because of base pairing, right? And finally then DNA polymerase will move along. DNA polymerase move along, joins up the bases that are slotted into the positions held in place by the hydrogen bonding. DNA polymerase joins them up by forming the phosphodiester bonds. Yeah, phosphodiester bonds, and if that occurs on both of the original 2 strands, we will end up with 2 new molecules, or 2 molecules of DNA, each containing 1 original strand and 1 newly synthesized strand. Okay, so again, the point of these videos is to avoid going into that detail. In fact, I will just remove that because I think that can be shown in a diagram much better. Okay, so what's important here is not it is important what we talked about, but we can express it in the diagrammatic form. So we have a molecule of DNA, this for example, okay, but it has to unwind and unzip, and that's the role of helicase. So that part of the molecule is not yet unwound, but helicase will come along and do this part of it. Yeah, and what happens is that the new strand is synthesized, but while one strand is synthesized continuously, so the DNA polymerase, so the free nucleotides will come along. Actually, let's just take it step by step. Free nucleotides will come along and align because of base pairing, and that will happen on the opposing strand as well, but they'll be that way. Yeah. Okay. So free nucleotides align any exposed bases, and then DNA polymerase comes along and catalyzes the formation of phosphodiester bonds on one strand continuously. Okay, but on the other hand, on the other strand, because it's the strand, remember, the anti-parallel, both strands are not conveniently going in the same direction. They have an opposite polarity. Because of that, it has to synthesize the other one in bits, in fragments known as Okazaki fragments, which will then eventually get joined up. So the separate fragments will get joined up by another enzyme. Okay, so the enzymes that are involved, and the third one is a bit of an extra thing, but the enzyme that's unzips and unwinds is helicase. The enzyme that forms the majority of the phosphodiester bonds is DNA polymerase, And then the enzyme that seals those tiny gaps left in the fragments on the other strand, because it couldn't be synthesized in a continuous way, is DNA ligase. Okay, so just be aware of those things. And now we will go on to Meselson and Stahl experiments. So I'm not going to explain this too much, though it is worth watching videos on Meselson and Stahl experiments at this point in your revision. Yeah, because examiners love applications of theory, and Meselson and Stahl is a good application of the theory. Okay, so Meselson and Stahl was all about bacteria grown in DNA. So bacteria would begin with one kind, with DNA that was lighter, actually heavy DNA, right? So they were grown in N15, right? So we'll indicate bacteria that have used N15 to build their DNA with black lines. All right, so we're taking these bacteria that have used heavy nitrogen to build their DNA, and we've transferred them into a growth medium that contains only light nitrogen. So any new DNA is going to have light nitrogen. Okay, so what would happen is, in the semi-conservative model, two strands would separate, new strands would be made, but the new strands would be made using lighter nitrogen isotope. And so we'd carry on, and if this carried on for one generation, it would look like this. But if we carried on for 2 generations, then that strand would separate from that strand, and so that strand would separate from that strand and that strand. And then we'd get the new strand, new strand, new strand, new strand. Okay? So if we analyzed this DNA in a tube, what would happen, right? So let's just draw it here. So if this was a tube, 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 and then you can spin the tubes down, the heavier the DNA, the further down in the tube it would go. Okay? So this DNA would be down here because it would be heavy, but this DNA, having 50% light nitrogen content, it would be higher up. So it's not that the color of what you'd see would change, but the band would be higher up in the tube. But here we'd have two sets of bands, because we'd have 50% of our DNA molecules would be completely made using the light DNA. So we'd have one band very, very high, but the other 2 molecules would be still one old, one new, so there'd be a middle height band as well. Okay, anyway, this is just me pointing to that. It comes up a lot, so just please prepare yourself for that. It's worth 5-10 minutes just watching a couple of videos on it for more in-depth explanation. So Meselson and Stahl, semi-conservative, replication, done. Let's move on. The other thing is protein synthesis. Okay, I'm going to try and do this quickly. Protein synthesis, remember, protein synthesis has 2 stages. The first stage is transcription. Yeah, and there are steps, but I don't want to go through the steps. And once transcription is done, we have translation. Okay, all I'll do is, at most, I will just draw a picture of what the hell's going on, and you need to remember how to describe it. Right? So in the nucleus, we have DNA. A bit of it unwinds, right? And instead of DNA nucleotides unwinding, so just think of it this way: in DNA replication, the whole molecule undergoes the process of unwinding and new strand synthesis. In transcription, only the length of one gene unwinds. Okay, and all of this happens because of the formation of the transcription factor complex or transcription initiation complex, a load of proteins all binding at the start of a particular gene. Why? Because signaling, right? At this point, I know this wasn't mentioned the first time you did protein synthesis, but at this point in your revision, you have to start putting a number of pieces together from different things that we've done. So, as a result of cell signaling, certain genes are transcribed, certain genes are expressed, right? Or during development, during differentiation, you've got gene expression changing in the cells to make them specialized. Well, that's happening because specific transcription factors are binding to the DNA now, but they weren't before. And why would that happen? Because protein activity is changing. Well, why would that happen? Because of cell signaling. So we've done that in other parts of the topic, and we need to start putting them together here. Anyway, as a result of cell signaling, hormonal, otherwise, certain transcription factors are binding to certain genes. When that happens, that gene unwinds new or RNA nucleotides this time will be lining up now that those bases are exposed. This time it wouldn't be DNA polymerase, however, that makes the phosphodiester bonds. It will be RNA polymerase comes along and joins up those RNA nucleotides with phosphodiester bonds, making an mRNA molecule that would move out of the nuclear pore into the cytoplasm, where it would encounter a ribosome, right? A ribosome, and remember the ribosome is binding to the mRNA molecule, and remember the ribosome might be on the rough endoplasmic reticulum because the protein that it's making needs to go into the membrane, or it needs to be released out of the cell by exocytosis, or it needs to undergo some kind of serious modification. Anyway, remember translation, the ribosome is reading 3 bases at a time, 3 bases at a time, called a codon. So we've got codons, right? 3 bases at a time, and remember this sequence of bases in the mRNA is complementary to the sequence of bases that was in the gene sequence of the DNA. Okay, so the gene sequence is very important because it essentially directs what the sequence of bases in the mRNA are. Okay, so mRNA. And the mRNA has got codons, and these codons will be read by the ribosome, and when a certain codon is in the ribosome bit, there will be tRNA molecules that will come along and have anticodons complementary to the codon of the mRNA. The job of the tRNAs, remember, is to deliver amino acids to the ribosome. So when these amino acids find themselves next to each other in the ribosome, brought next to each other because of the tRNA molecules, and the tRNA molecules, remember, are there because the codon on the mRNA says so, and because they're complementary, right? And when the amino acids find themselves next to each other, they get joined by a peptide bond because the ribosome is helping to catalyze a condensation reaction. So this is also connected to protein structure that we did very recently. Yep. And as the ribosome moves along, the peptide chain gets longer and longer and longer, okay, as more amino acids are added on, codon by codon, along the mRNA. All right, and thus we have the beginnings of a polypeptide chain, which will eventually have to undergo folding, secondary structure, tertiary structure. It might need to combine with other polypeptide chains to form quaternary structure. We might need to stick something non-protein on there as a prosthetic group, but then that protein will work and do stuff in the cell, causing the cell to do biological things, make responses to hormones, for example, or catalyze other metabolic processes, producing other biological substances necessary for the body. All that stuff, okay? So a lot of what's going on here is underlying a lot of other biology, is what I'm trying to say. Right, so we have translation, we have transcription, and so we produce proteins. So let me just put that right there. We will make a protein or a polypeptide, and that protein will have various functions. It might have functions in the cell, it might have functions outside of the cell. But that's very important to appreciate, that we have just made a protein, and protein are central to a lot of what's going on in biology. Yeah, right, protein synthesis, transcription, translation. Here I'm just going to add a little bit to do with genetic code. So protein synthesis, I'll just put it here, in fact, that's a nice place, right? The properties of the genetic code. You don't have to remember all the codons, but remember they'll give you a table, and you might have to read off the complementary bases, or which bases give rise to which amino acids, and so on. But it's important to understand the genetic code. Actually, protein, I missed something here, very, very important. Translation is where the amino acids get put together in a particular order. That is primary structure. Okay? And if the primary structure is fine, if all the amino acids are in the right order, then the protein will have the right shape and it will have the right function. But if the primary structure is wrong because of wrong codons, because of mutation in DNA, then the function might be not quite right. Okay, so it's important to understand how mutations and everything fits into that story. So the genetic code has a certain nature to it, right? I think there are certain things mentioned on the specification which I'll just need to refer to. Right, genetic code. Few things mentioned on the specification. First, triplet. Yep, 3 bases make up each unit of information in the gene. Okay, so triplet, 3 bases. Not 4, not 2, 3. Right, next, triplet, what was the next thing? Non-overlapping. Non-overlapping, i.e. it's 3 bases and then it's the next 3 bases. It's not these 3 bases and then the 2 of the first 3 bases and then the next. Yeah, so it's 3 bases, then the next 3 bases, then the next 3 bases. The 3 bases don't overlap with each other. Triplet, non-overlapping, degenerate. Degenerate. Degenerate means that more than one codon can code for the same amino acid, not the other way around. Okay, so degenerate means you can have a number of different codons that might code for the same amino acid, which means that there's a little bit of give, right? So sometimes mutations don't result in an altered primary structure, because even though we've changed a codon, that's the different codon, though the new codon still codes for the same amino acid. Finally, universal, right? Different organisms don't have different interpretations of the code. They have the same, right? At least on this planet. So, universal, it is. Again, there might be other things to do with that. The best thing to do with this, though, try exam questions. And also, find Meselson and Stahl exam questions. Find good application questions that connects to these things, and I think that would be good preparation. All right, extracting DNA. Extracting DNA. So, I think the main thing to think about when looking at extracting DNA is to think about where you'd find it. It would be in the nucleus, right? So you've got the DNA, and it's in the nucleus of the cell. So how would we get to it? And once we've got to it, how do we remove the other stuff that we don't need? Okay, so one, we treat with well, we might mash up the cells first to increase the surface area and so on, but once we've done that, we would add detergent. And what the detergent does is, as we've looked at membranes already, right, detergents disrupt the membrane structure, allowing the DNA to come out of or escape the membranes, the nuclear membrane or the nuclear envelope and the cell membrane. Okay, right, we would filter it, because now that we've broken open the cell membranes, there might be quite a lot of cell fragments kind of floating around, and we want to separate the DNA from that. So once we've separated that, then once we've filtered, we would add salt. Right? Now salt, it helps the DNA to separate from the water molecules. Next, we would add proteases. Remember that the DNA in a cell, the DNA is associated with histone proteins. Right? Now, for most purposes that we want to use DNA, we don't want the proteins, we just want the DNA. So we treat with protease enzyme, and that kind of breaks down the histone proteins that the DNA is associated with, leaving the DNA by itself. 5, we would add cold alcohol or ethanol. Right? And what this does is it precipitates the DNA, because so far the DNA is kind of dissolved in the water. But when we add cold alcohol, the DNA precipitates out. So this causes a precipitation of the DNA, which means that it's no longer dissolved with everything else. Okay, and once it forms a solid precipitate, we can then physically remove it. We can even give it a rinse a few times to make sure that all the stuff that's not supposed to be there is removed. Okay, so we have that. But anyway, it's important to know, if you're not satisfied with that very quick discussion of that, look at the steps. It's important to know the steps and why each step is done. Okay, right, that takes us to ATP function, isn't it? Could I be cheeky and just draw the ATP function going up there? Okay, so ATP, remember that ATP, these phosphate groups are negatively charged. So you put 3 phosphate groups, each with their negative charge right next to each other, that is going to be quite unstable. Okay, that's going to be quite unstable. So ATP is an energetically unstable molecule that we've put energy in to make it. Okay, so ATP is kind of interchanging with ADP. So we've got a phosphate group by itself, and we've got 2 phosphate groups, and we could have ADP. That's ADP, and that's ATP. Okay, now this is a much more energetically stable situation, right? Yeah, because we've not jammed 3 negatively charged groups into close proximity to each other. So this is much more energetically favorable, and ADP is a much more stable molecule. But what we could do is put energy in. We could put a lot of energy in. Yeah, energy in. So for example, let's say we've released some energy from the breakdown of glucose. We can put those energy in small bits into making an ATP molecule, which is a temporary store of that energy. And then later, when we need the energy, we simply convert ATP to ADP, and that releases a small packet of energy that we can use to do active transport or something else. Okay, so just remember, ADP, more stable, but it's lower energy. More stable, lower energy, and ATP is essentially less stable but high energy. Okay, and ATP is a small molecule, so it's polar. Mostly polar. It's easily dissolved. It releases energy in kind of small amounts, rather than kind of if you released all the energy in glucose all at once, it would be a lot for the cell. Rather, ATP is releasing small amounts of energy. Okay, so I would say just have a quick look at ATP. Structurally, you get what it is, but understand how its structure connects to its function. That might be a useful thing to do. But apart from not going into that detail too much, I think that is nucleic acids. Right, on to the next one. Just forgot something. Actually, a couple of things. I think when we did this, I think it might be useful if you made a little table here, just comparing all the different types. So DNA, obviously, like what role did DNA have in transcription and translation? So make a few points about how DNA is involved in this process. Secondly, RNA. Now, RNA has got a number of different forms. So it's involved at a number of different stages, but it's also involved in different forms. Look at this. RNA is involved when it's produced as mRNA, number one. 2, RNA makes up the tRNAs. Okay, so have an awareness of tRNAs, general structure, function, etc. And finally, RNA is also forming the ribosomes. So something which we call rRNA. So ribosomes are a bit of a, they're a lot, just RNA. They're RNA-based structures. There's a bit of protein in there, but primarily ribosomes are RNA. So just be aware that RNA has got a lot of involvement in protein synthesis, but it's not just because of mRNA, it's the tRNA and the ribosomal RNA as well. Okay, the other thing that I forgot to mention, but which comes up quite a bit, is being able to compare and contrast DNA and RNA. Okay, so I will just put it here, structure, compare. Now that doesn't mean just differences, it means be aware of structural similarities and structural differences. So the fact that they both have phosphate, they both are made of nucleotides which both contain phosphate, pentyl sugar, nitrogenous base. Similarity, done. But what are the differences? For example, DNA has deoxyribose as the pentyl sugar, RNA has ribose as the pentyl sugar. We've looked at it, but it's just to kind of be able to put it in a direct comparison context. Okay, so compare, and that means similarities and differences. All right. Similarly, we can compare functions as well. Yeah, we can compare. Yeah, I mean whatever. Yeah, so you might be asked to compare ATP to RNA, for example. So you might be able to say that, well, they both have ribose as their pentose sugar. However, RNA can be a polynucleotide, ATP is just one nucleotide. You know what I'm getting at there. Okay, so be able to compare functions, compare and contrast with functions, compare and contrast with structure. And I think that that is now finally that. Good luck.